All right, so any questions before we get started? Today we're going to cover genetic disorders. We'll cover cancer genetics, genetic testing. Genetic testing is too much information for one session, so we've kind of pulled out, excuse me, next generation sequencing into a session all by itself. And what we're going to do here in the next generation sequencing session is we'll have a case where next generation sequencing identifies millions of variants, and we're going to have to sort through those variants to try to figure out which of them are significant and which one might actually be the culprit mutation causing disease in the individual. Genetic engineering, there's a lot of genetic, in every one of the sessions we try to talk a little bit about some of the treatments, and many of them these days fall back to genetic engineering, and we'll wind up talking a little bit about genetic engineering today and next week as well, but then we'll really try to pull it all together and do a deeper dive into the genetic engineering during this session. Okay. So for today, we're going to just talk about the reminder about the homework assignment for the ethics class. And then we're going to give an overview of genetic diseases and talk specifically about two diseases because they illustrate broad classes of genetic diseases. And then we'll talk a little bit about molecular treatments. Just kind of categorize the various molecular treatments that are out there. Okay. And this is the same homework slide we've been looking at for the last couple of weeks. This is just a reminder that topic 12 coming up in July, if you guys could begin to think about these topics. By the way, I'll give individual feedback to everyone for your homework assignments. Thanks for sending in your homework assignments. You guys really did great. Everyone's going to get an A, an A+ on the homework assignments, and but I'll be sending each of you individual feedback on your homework assignments. You guys did a really great job. Okay, ready to start? Okay, good. I'll take that as a yes.
Yes, Bob.
So, okay, thanks. So, let's begin with an overview of genetic diseases just by trying, and it's really difficult, any kind of categorization you might choose to use is going to have some gray areas and places where it doesn't completely reflect all of the different types of genetic diseases, but the categorization you see here is pretty good. What we'll see with genetic diseases is abnormalities that alter the protein or alterations that have a normal protein structure but change the amount of the protein or allow that protein to be expressed in the wrong cell, for instance, or at the wrong time. And we've seen examples for many of these, and we'll see more examples as we go forward, including today. And so, for instance, most of the mutations we've looked at for like sickle cell disease, some of the thalassemia mutations, and we'll also look at cystic fibrosis today, result in an abnormal protein. Rarely, the abnormal protein has a novel property. The protein keeps doing what it's supposed to be doing, but on top of that has acquired some new property. And we saw that with sickle cell disease, where the sickle cell hemoglobin continues to ferry oxygen back and forth the way it's supposed to, but the novel property introduced by the mutation was the propensity for sickling under low oxygen tension. We'll see cases where an abnormal protein actually leads to a gain of function. We'll come back to this a little bit next week when we talk about genetics of cancer, and we talk about this notion of oncogenes, or genes that are, when they're mutated, they result in a gain of function, actually, for instance, stimulating the cell cycle to further divide. And we also have seen where the protein is abnormal, we have a loss of function, and we'll see this again today for cystic fibrosis. And by far, this is the majority. Of genetic diseases, at least the straightforward monogenetic disorders, is we have this notion of a point mutation, and that changes the protein structure, and as a result, the protein function is diminished. We've also looked at some diseases where the protein structure is normal. And we saw this, for instance, when we looked at thalassemia, and we saw the hundreds of different mutations of the hemoglobin beta gene, where some of those mutations were, for instance, in the promoter region and led to decreased transcription of the gene. And the thing with thalassemias and with the hemoglobinopathies, you can almost always look and find examples of all kinds of different diseases because of the varied mutations. So we've seen examples where you have a decreased amount of a normal protein. We saw that with thalassemia. There's examples where you have an increased amount of a protein. And we see this, for instance, where you have large gene duplications, such as in trisomy, such as in Down syndrome, for instance, where the genes are normal, but you just have an extra copy of the gene, and having that extra copy of the gene results in an increased amount of the protein product, and that leads to disease. And then we'll see other cases, and we'll see this again next week when we talk about oncogenes, where the protein is normal, but it's expressed at the wrong time or the wrong place. And we'll see a lot of examples where the cell cycle is finely tuned, but in cancer, when you have certain genes being expressed at the wrong time, you have then overactivation of the cell cycle with uncontrolled cell growth. So this is kind of a broad way of looking at the spectrum of genetic diseases. Another way of looking at the spectrum of genetic diseases is to kind of break them down into three large buckets. One of the buckets that we've looked at are the disorders due to single gene mutations. This would be, for instance, like in sickle cell disease or cystic fibrosis, where you have a point mutation, and that point mutation alters the protein, and the altered protein then results in the particular phenotype. Disorders due to genome and chromosome mutations are generally those diseases where the gene itself is just fine, but you have extra copies or lesser copies of the gene. So, for instance, an example of this would be in Down syndrome, trisomy 21, where the genes on chromosome 21 are all normal. There's no mutations in the genes, but you have extra copies of the genes. So this is also referred to as a dose effect. And so there'll be this category of disorders where the genes themselves are normal, but just because you have more copies of those genes, you'll wind up getting the disease. And then by far the largest category of genetic diseases where there's tremendous activity and study going on is these disorders with multifactorial inheritance. We'll talk specifically about type 1 diabetes today, but there's just a whole boatload of diseases with multifactorial inheritance. Common diseases, schizophrenia, autism, hypertension, diabetes, where people have talked about nature versus nurture. We see a higher incidence of these diseases, a higher clustering of these diseases in families, but there's no single gene responsible, and there's no clear-cut Mendelian inheritance pattern for these. And by far, these are the most common, and so we'll start examining one of these in particular today when we look at type 1 diabetes. So finally, by way of overview, we can say there's really these three main types of genetic diseases. And again, all of these categorizations have areas where they break down, but this is, I think, a convenient way of kind of conceptualizing genetic diseases. We'll be looking specifically at an example of a monogenic disorder today, cystic fibrosis. And we'll be taking a detailed look at a multifactorial disorder today, type 1 diabetes. The other thing I wanted to try to do today was to kind of take you through the whole process that went from, okay, you take cystic fibrosis or you take diabetes, and at some point, someone came to the realization that these were genetic diseases, okay? So how was it that people came to the realization that these are genetic diseases? And having determined that these were genetic diseases, what's the process that folks have gone through in order to actually isolate the gene and isolate the actual mutations? So today, I thought it might be interesting to not just go into the genetics of cystic fibrosis or diabetes, but also take you through a little bit of the exercise that went on to determine that it was a genetic disease and what the genetics of the disease actually were. And there's three common methods that are used for gene/mutation detection. And today we'll be looking at the gene detection method called linkage. And we'll be looking at that in the context of how we figured out the genetics of cystic fibrosis. And we'll be looking at the detection methods of association, specifically with respect to diabetes.
That's all right.
Excuse me? We'll come back to the DNA sequencing method in detail when we come back to the next generation sequencing scenario. So for today, we're going to look at how was the gene/mutation for cystic fibrosis detected by linkage, and how were the genes that contribute to diabetes determined through genome-wide association studies. Okay. Any questions before we go on? Okay. You guys are a quiet group today. Okay. So far, so good? Everything making sense? Okay. Can you guys still hear me?
Yes. Yes.
Yep.
Yes.
Okay. I'm trying to get you guys to talk a little bit. Okay. All right. So let's begin with cystic fibrosis. And this is a unfortunate kid that died of cystic fibrosis, and this is his lung in cross-section. And you can see the mucus plugs in here and the purulent secretions plugging his airways in here. And so he died as a result of cystic fibrosis that just destroyed his lungs. So JB is a 2-year-old. He was referred to the clinic for evaluation of poor growth. And this 2-year-old, when he was an infant, had diarrhea and colic, but it resolved when an elemental formula was substituted for a standard formula. And then as table foods were added to his diet, he developed malodorous stools containing undigested food particles. During his second year, he grew poorly. He developed a chronic cough. He had frequent upper respiratory infections, but no one else in the family had any of these problems. His weight and height were plotted less than the 3rd percentile, and his head circumference was at the 10th percentile. When he was examined, he was found to have a severe diaper rash. His lungs were just filled with all kinds of sounds, all kinds of bronchi. His fingertips looked like little clubs. Clubbing of the fingers is often seen with advanced lung disease. And the pediatrician suspected that he had cystic fibrosis and ordered a sweat chloride test and found that the sweat chloride level was very high, consistent with cystic fibrosis. And so this patient was then referred to a cystic fibrosis clinic for further counseling, mutation testing, and treatment. And generally these days, people with cystic fibrosis have their DNA sequenced because, as we'll talk about, when we talked about sickle cell disease and when we talked about thalassemia, it was kind of like, well, if this gene is mutated, then you wind up having a phenotype, you wind up having a disease, and therefore there's a treatment. But with cystic fibrosis, we're starting to kind of dip our toe in the water of precision medicine, where the treatment is not based purely on the fact that the gene is affected, but rather the specific treatment that's used for cystic fibrosis is really dependent on the specific mutation. If you have a mutation over here in the gene, you're going to get one kind of treatment. If you have a mutation over here in the gene, you're going to get a different kind of treatment. And so we'll take a look at the specifics of treatment and how they relate to the specific genetic changes that are seen in the disease. And here's some other pictures of cystic fibrosis. You can see the lung over here with how it's swollen with the mucus plugs. Here on the left, you can see the pancreas. And you can see in a normal pancreas up here, you can see normal pancreas cells, the normal islets, the insulin secreting areas of the pancreas. And on the bottom left, what you see is a pancreas that's been largely replaced with fat and scar as a result of the cystic fibrosis. What's going on in cystic fibrosis is that the gene that produces the protein is mutated, and the cystic fibrosis transmembrane regulator, the CFTR gene, produces this protein, and this protein is responsible for sodium and chloride exchange across the epithelial membrane of sweat glands or lung epithelium or gut epithelium or pancreas epithelium, and it controls the amount of sodium and chloride in the fluid and indirectly thereby controlling the amount of water content or the hydration status of the fluid. And so here in the normal situation on the left, where you have a normal CFTR protein, the amount of sodium and chloride being absorbed results in hydration of the liquid layer, and here, okay, okay. So anyhow, here on the left, here on the right, where we have a mutation in the gene, and as a result, we have a defective protein. What happens is the amount of sodium that actually is being absorbed increases. And the reason for that is that the CFTR receptor actually has an inhibitory effect on another transmembrane receptor, the epithelial sodium channel. And so as a result of the loss of the inhibitory role of CFTR, you have increased sodium being absorbed from the lumen, and the water passively follows the sodium, and you wind up getting dehydrated, viscous, thick secretions in the airways, and then these are prone to being infected with organisms like Pseudomonas, and that leads to a lot of the pathophysiology of the disease. So the role of this receptor, the role of the CFTR gene product is really to control sodium and chloride transflux across the epithelial membrane, and as a result, also control the hydration status of the mucus layer. And when the gene is mutated and you no longer have the receptor there, you wind up, in the case of the respiratory epithelium, getting these thick secretions that then lead to the phenotype of the disease. Cystic fibrosis is an autosomal recessive condition, and we looked at these pedigrees last week, and I'm just showing you again some typical pedigrees for cystic fibrosis. I think we even looked at the frequency of these mutations, and so oftentimes what will happen is a couple will marry, neither of them know that they were a carrier, but they'll wind up having a couple of kids with cystic fibrosis, and then from there we'll discover that both of them were carriers of the cystic fibrosis mutation, or at least of one of the cystic fibrosis mutations. Some of the time, sometimes what you'll see in the pedigree is a consanguineous mating, where the mother and father really are kind of directly related. And in these situations where you see an affected offspring with a disease, that's often a clue that the disease in question is the result of an autosomal recessive inheritance pattern. So let me just back up a minute. So, okay, healthy baby syndrome, cystic fibrosis, it's been recognized for a long time. How was it that people, how do you think, what led people to conclude that cystic fibrosis was a genetic disease? What do you think may have led people to believe that cystic fibrosis was a genetic disease? And don't think too hard, because the answer is pretty obvious. You know, what would make you think that cystic fibrosis is a genetic disease?
It's early detection.
Well, the fact is it clusters in families, and when you build family trees, you'll see that you look at this pattern here, and nowhere was there any cystic fibrosis, but boom, we just had 2 kids that have cystic fibrosis, right? Or you see in a consanguineous mating, you have a child born with cystic fibrosis. And in most of these cases, the same thing with diabetes. You know, what is it that leads someone to believe that a disease has a genetic basis, whether a pure Mendelian basis or, you know, possibly a partial genetic basis of a complex multifactorial disease? And the answer is going to be, well, you look at the pedigree. or you look at a lot of family histories or a lot of family trees, and in these family trees, when you start seeing clustering of diseases in families, if you saw this, if the patient came to you and said, I have cystic fibrosis, or I just had I had 2 kids with cystic fibrosis, the lights would start to flash and you begin to suspect that this might be a genetic disease, okay? And so here again, with a consanguineous mating, we see a child with cystic fibrosis. That's a clue not only that cystic fibrosis is a genetic disease, but also that it's an autosomal recessive genetic disease. So initially we're clued in from the family history or multiple family histories that a particular disease is a genetic disease, and from there, having concluded that this is probably a genetic disease, the next question is, okay, but where is the gene for this disease, and what is the mutation for that disease? And for cystic fibrosis, the technique that was used is a technique called linkage analysis. And let me see, I'm going to try to walk through linkage analysis. I'm not going to give a very mathematical description of linkage analysis, but rather what I'm going to try to do is give like a conceptual overview of what linkage analysis is. And really, in order to do that, and it's pretty simple, it's really just remembering what happens in recombination and recognizing that if 2 different spots on a chromosome are far apart, then there's a chance that crossing over will occur in between those markers, whereas if markers are very close together, there's less of a chance that crossing over will occur between them. So crossing over is more likely between spots that are far apart. And here, where we see A1 and B1 sticking together, we can say that those spots or those loci are linked. Or another way of saying it is that they're in linkage disequilibrium. Back when Mendel described the classic Mendelian inheritance patterns, he described the 3 laws of Mendelian inheritance, one of which was independent assortment, which is, whether the P's are wrinkled is completely orthogonal to whether or not the P is yellow. But here we see, if you will, a divergence from Mendelian's law of independent assortment, in that if the markers that we're interested in at the A spot and the B spot are close together, if one child inherits allele 1 at marker spot A, he's also likely to receive allele 1 at marker B. Whereas if these markers are far apart, or if these markers are on different chromosomes, then what we'll see is closer to Mendel's law of independent assortment, where one child might receive marker allele 1 at marker A, but because crossing over occurred, or because they were on separate chromosomes altogether, may have received allele 2 at marker B. So really the thing to notice here is that the closer these markers are, the more likely they are to be inherited together, the less likely there's going to be a crossover event in between them. The more tightly linked we say these are, or we say that there is linkage disequilibrium between these markers. Okay. Now, the other thing about this picture is locus B might be the gene responsible for the disease, but we may not have the ability to directly, especially, back in the 80s when folks were trying to discover the locus for cystic fibrosis, we may not be able to directly measure alleles at spot B, but we may have the ability to measure alleles at locus A. And if we see that every time a family member that has allele 1 at marker A also has cystic fibrosis, and every time the person has allele 2 at marker A does not have cystic fibrosis, that then is compelling mathematical statistical evidence that the gene for cystic fibrosis must be very near the marker for A. It must be in linkage disequilibrium with the marker we're studying. And if we look at another picture, I know this is kind of small, I hope you guys can see this okay, but this is an example of a family tree showing how linkage disequilibrium allows one to narrow down the region where we might find a causative gene. And it allows us to narrow down the region. It is not necessarily going to tell us exactly what the causative gene is. It really depends on the density of the markers. And back in the 80s, we had maybe 1 or 2 orders of magnitude less markers than we have available today. And so we could get kind of a gross narrowing down of the region where the cystic fibrosis gene was. So here we have a pedigree where you have a male that has a disease, and a female that doesn't have the disease. And we've been able to measure markers at 5 different spots on this particular chromosome. The children, the female offspring, also has disease and inherited chromosome from mom and a chromosome from dad. And then that female has children, but what's interesting to note is that 2 crossover events occurred in the children. In this crossover event, in this patient here, we see that she now has homozygosity for marker A1, but still has B2 through E2. And over in this patient over here, there was a separate crossing over event. And so based on the study of the markers, while we don't necessarily yet know where the culprit gene is, we've been able to narrow down the region. The recombinants localize the disease-causing gene to the region between markers A and D. We know that it's not up above A because we would have seen it during one of these crossover events, or likewise below. And so this kind of illustrates how we can use linkage analysis, linkage disequilibrium, the fact that genes that are close together have infrequent crossover events in between them to begin to narrow down where the culprit gene is. Ultimately, the gene for cystic fibrosis was found to be on chromosome 7. It's about 200,000 bases long. It has 27 exons, and it's got hundreds of known mutations at this point. And we'll come back to this again to look at the specific mutations because, again, the different mutations result in different patterns of disease and therefore result in and actually are amenable to different types of treatments. Okay. When we look at the cystic fibrosis gene, depending on where the mutation is, you'll see different phenotypes. Folks have tried to categorize the different mutations into different classes. And sometimes the treatments for cystic fibrosis are class specific, although sometimes the treatments for cystic fibrosis are actually mutation specific. So we have the class 1 mutations where you're simply not producing any protein whatsoever. You have the class 2 mutations, which are the most common. And the class 2 mutations are you produce some protein, but the protein is improperly structured so that it doesn't work its way. It just gets dissolved ultimately in the Golgi apparatus, and it doesn't work its way all the way to the epithelial, to the membrane. Class 3 mutations do actually insert themselves into the membrane, but they're not fully functional. Class 4 mutations, don't insert themselves into the membrane. The class 5 mutations are usually RNA, messenger RNA transcription mutations that result in a decreased amount of protein being produced. And then we have class 6 mutations where we have accelerated turnover. So you have a receptor that inserts itself into the plasma membrane, but then quickly gets retaken back into the cytoplasm and digested. And looking at this in another way, here's the CFTR gene. The most common mutation is a mutation where there's no phenylalanine. The phenylalanine at position 508 in the protein is missing. The proteins, so here's the cell membrane here, and we can see the protein, and it has these membrane spanning domains, and it has these other domains for achieving its function. And mutations anywhere along the chain can be responsible for different classes of mutations. We see the exons here. So again, the different types of mutations that are described for cystic fibrosis have been broken down into 6 classes. The class 1 are the absent proteins. Class 2 is significant for a couple of reasons. One is because this is the most common mutation where there's simply a major block in the protein maturation. The protein is produced, but it's degraded in the Golgi apparatus, and it doesn't therefore serve any function whatsoever. Another class that's very important because there's now some treatment available are these class 3 mutations called defective gating, where the protein actually does insert itself into the cell membrane, but it's not highly functional. It's marginally functional. And here's an example of one of these where at position 551 in the protein chain, the glycine is replaced by an asparagine. And I highlight this one, and I highlight this one in particular because when we look at various treatments, we'll see that there's a specific treatment for this specific mutation. There's a specific treatment for this specific mutation. There's also specific treatments different from the treatments for these mutations, for the class 5 mutations, which result in reduced expression of the gene, at least for some of the class 5 mutations. And then class 6 mutations are again illustrated here where the protein is produced. It makes its way to the cell membrane, but then gets rapidly taken back into the cytoplasm, so it's unstable. So if we talk a little bit about treatment and we start to broach the subject of personalized or precision medicine, I think this slide nicely illustrates that concept where, again, we're not just treating cystic fibrosis, but we're treating this specific mutation of the gene. we're not just treating the fact that the gene is mutated, but we actually are targeting the treatment based on the specific mutation. And so mutations, class 1 mutations, may have different treatments than class 2 mutations, which may have different treatments from class 3 mutations, although even within a class, the treatments that are being explored can vary. So here again, this is the most common mutation, is the absence of a phenylalanine residue at position 508. And this particular mutation, the messenger RNA is transcribed, but the protein that's produced is an incorrect protein, and therefore it gets destroyed in the Golgi apparatus and it never makes its way to the cell membrane. Versus these class 3 mutations, where we actually do see a protein makes its way all the way to the cell membrane, but it's not highly functional. It's marginally functional. And so we have specific treatments now that work for some of the class 2 and some of the class 3 mutations, as well as treatments that are targeted against other classes, such as the class 5 mutations, where the mutation results in decreased transcription. And so just kind of summarizing the different types of treatments, here's a couple of articles that may be of interest to you. And there was a lot of hype about the release of ivacaftor because it was the first time we saw that a molecular treatment could be effective in a genetic disease. And the history of ivacaftor is kind of interesting. The company screened thousands of small molecules, and they found that ivacaftor resulted in a conformational change. This particular mutation, the glycine 551 is replaced with an asparagine, is a class 3 mutation, a class 3 mutation where the protein is produced. It actually works its way up into the plasma membrane, but doesn't work very well, and it's not very efficient at transferring chloride ions. And so the ivacaftor somehow inserts itself within the protein, changing the three-dimensional conformation of the protein, allowing it to actually become an effective channel for chloride ions. It doesn't completely restore the function, but it does partial restoration of the function. And so now it's FDA approved, it's standard that if somebody with cystic fibrosis, and about 10% of patients with cystic fibrosis have this particular mutation, those patients that have this particular mutation can now be treated with ivacaftor. The more common mutation, the absence of the phenylalanine at position 508, there's another drug, lumacaftor, which is given in combination with ivacaftor. Although the success isn't as great, and the jury is still out on the lumacaftor, but this also changes the conformation of the protein, and together with ivacaftor, appears to bring it to the cell membrane where it can effectively channel chloride ions. And then we have a lot of other compounds under development, and we'll come back to this when we talk about molecular treatments of genetic disease a little bit later today, and we'll come back to this again when we get to genetic engineering. But just to give you an idea of some of the other types of molecular therapies being developed, either for cystic fibrosis or for a number of other diseases, there are drugs that can facilitate premature stop codon read-through. So for instance, you get a point mutation that results in a normal codon being changed to a stop codon. There are drugs that will facilitate the ongoing transcription past the point of the premature stop codon. And here where you have, for instance, class 5 mutations in cystic fibrosis where you have less protein, or you have a class 4 mutation, or you have no protein in class 1 because of a mutation that results in a stop codon, then these premature stop codon read-through drugs may have an effect. We have something called antisense oligonucleotides that alters messenger RNA transcription. And in some of these cases, and we'll come back to this one as well, the antisense oligonucleotides get into the nucleus and they bind to the DNA and they alter the transcription pattern. So for instance, where there's a mutation in an intron that results in abnormal splicing, the antisense oligonucleotide can override that and lead to normal messenger RNA transcription. And then we have gene therapy, which is also something we'll come back to, which is introducing corrected genes into the cell. So it's a nice example, and it's a nice introduction, I think, to this notion of precision medicine, where it's not just enough to know that the patient has cystic fibrosis, but it's important to know what the specific mutation is so that the therapy can be targeted. So, disease-causing mutations most commonly cause disease by A, B, or C. You guys still out there?
Is it a loss of protein function?
Loss of protein function. Yep, that's right. Okay, so by far most common, we see loss of protein function. And number two, which type of genetic disease is the most common? The single gene, the multivectorial, or the chromosomal triploidy and segmental duplications?
Multifactorial.
Good, good, okay. Now here's a tough one. You guys are doing, you're doing too well on the easy ones. Okay, so most patients with cystic fibrosis have a particular allele at locus D7S23. Okay, now it just so happens that this is a marker that in the 80s we were able to study. And we found that, look at this, patients with cystic fibrosis generally have a particular allele at this locus. Okay? So a particular allele at this locus was commonly seen along with cystic fibrosis. We knew that this marker was on chromosome 7 in the 31 to 32 band. So this question is getting at, what does that tell us? Does it mean that this particular locus is in the causative gene? So what we're seeing here is that this particular marker, that a particular allele of this marker is seen when we see cystic fibrosis. Does that mean that this marker is actually in the cystic fibrosis gene?
No, it means there's locage disequilibrium.
Right, that's right. It could be in the gene, but it's not necessarily in the gene. This locus could just be very close to the gene so that there's linkage disequilibrium with the causative gene. Kind of sort of makes sense? No? Yes? Kind of, sort of? I think, a real mathematical discussion of linkage disequilibrium we're not going to cover that, but hopefully the conceptual notion that you have a locus that's near the disease-causing gene, and if you can look at alleles of this particular locus, and you see that certain alleles co-occur with a higher than random chance with the cystic fibrosis, then that tells you that the cystic fibrosis gene is probably near this particular locus because of linkage disequilibrium. Kind of sort of makes sense?
Yes.
Okay. Okay. You guys are a quiet group today. Okay. Let's switch to diabetes because linkage disequilibrium is difficult and not necessarily as interesting to talk about as the genetics of the immune system. And so when we talk about diabetes, particularly type 1 diabetes, and particularly about the autoimmune aspects of diabetes, I find it just out of this world fascinating to consider the immunologic aspects and to study how genetics influence our immune system. This particular picture is a guy that at the age of 28 was diagnosed with type 1 diabetes or insulin-dependent diabetes. And this picture on the left shows him after 3 weeks of polydipsia and polyuria. And then here on the right, he's put on 5 kilograms after he's been on insulin replacement. The case we're going to talk about is a 45-year-old guy with late onset diabetes. And he was referred to the genetics clinic for counseling regarding his children's risk for diabetes. So you got a 45-year-old guy, just comes down with diabetes, and now he wants to know what about his kids, are they at risk for diabetes? The patient developed glucose intolerance at the age of 39, and then fasting hyperglycemia at the age of 45. He did not have a history of any other problems. He was slightly obese. His body mass index was 31.3, and he had 5 children. He had 5 children by 2 different partners, and a child from each relationship had developed type 1 diabetes. So right there you see, okay, this guy has diabetes, he's got 2 kids from 2 different wives, each of them have diabetes, and they developed their diabetes before the age of 10. So right there you begin to think to yourself, okay, looks like we're dealing with a genetic disease. His sister developed diabetes as a child and died during adolescence from diabetic ketoacidosis. And the geneticist explained that the patient appears to have a late onset form of type 1 diabetes, and that his non-insulin dependent diabetes was probably an antecedent to the development of insulin dependent diabetes. In other words, his pancreas was just kind of pooping out. So it looked initially like he had type 2 diabetes or non-insulin dependent diabetes, but ultimately he manifest insulin-dependent diabetes. And so the patient enrolled himself in a research protocol, and as part of that study, him and his children were tested for anti-islet antibodies because it's thought that autoantibodies, antibodies directed against the islet cells of the pancreas, the insulin-producing cells of the pancreas, contribute to the pathophysiology of diabetes. And he was found to have antibodies, and one of his unaffected daughters had a high titer of antibodies. And that daughter also had an abnormal glucose tolerance test, but not fasting hyperglycemia. and so they were started on the insulin. So this looks to be a family history of diabetes, and just for fun, I put in this picture showing diabetic retinopathy, at least early non-proliferative diabetic retinopathy, where you can see here some small hemorrhages occurring, and you can see some cotton wool spots, and you can see some hard exudates in the retina of this person with diabetes. And most of you guys are clinical and know all of this stuff, but anyhow, a couple of slides just to remind people about diabetes. Here are the excuse me, criteria for the diagnosis of diabetes, which is basically if your blood sugar is high. And a difference between type 1 diabetes and type 2 diabetes. Usually with type 1 diabetes, you see it has its onset in youth. The other thing you'll see in diabetes is the monozygotic twin concordance is higher than what you'd expect if this were not a genetic disease. So in type 1 diabetes, if one child has diabetes, the risk of the other child having type 1 diabetes is 1 to 6%. And if one member of a monozygotic twin pair has diabetes, then the other twin, the risk of them getting type 1 diabetes is 35 to 50%. So it appears, even though the pedigree doesn't show this to be a classic Mendelian pattern, it appears from sibling studies and monozygotic twin studies that there is at least a genetic component. And this is typical for what's seen with multifactorial diseases, is that the pedigree doesn't reflect a clear-cut Mendelian inheritance pattern, but we see that there is still a statistically elevated clustering of that disease in families. And again, when we look at the risks for diabetes, we'll see that the risk for developing type 1 diabetes in the general population being 0.2, but in monozygotic twins it's 40%, in siblings it's 7%. And so when we look at these numbers, we begin to say, okay, it looks as though there is at least a genetic component to diabetes. Maybe not a classic Mendelian inheritance pattern like we saw with cystic fibrosis, but at least there is some type of possible multigenetic influence going on here. And so the question is, given that we now believe there's a genetic component to diabetes based on these family studies, how are we going to tease apart what the genetics are? And in trying to isolate the genes responsible for diabetes, we'll be looking at association studies as opposed to linkage studies. And so we used linkage studies. We looked at linkage for cystic fibrosis, where whenever the person had the A1 marker, they also had the disease. In association studies, really at a high level, just kind of conceptual level, you have these large databases, and you can say, okay, here's a group of people that don't have diabetes, and here's a group of people that do have diabetes. Okay, what do we see? In the group of people that do have diabetes, is there a statistical increase in any of the alleles over the controls? And so the association studies are really a statistical exercise where we're looking for a statistical increase in the frequency of genetic markers in the cohort of people with the disease compared to those that don't have the disease. And these genome-wide association studies are particularly useful where you have these complex polygenetic disorders in trying to isolate at least a spot next to the causative gene, or actually, depending on where your markers are, you might actually find a spot within the gene itself. And when these association studies were applied to diabetes, one of the things that was discovered was that a couple of genes, actually a couple of alleles at a couple of genes, were found to be more prevalent in the diabetic population than in the non-diabetic population. And there's a number of genes that were found to be, well, actually a number of alleles that were found to be associated with type 1 diabetes. A number of different genes and a number of different alleles in those genes, but in particular, the strongest association that was found were in genes DQB1 and DQA1, and these are genes that code for HLA antigens. Okay, we've talked a little bit about the major histocompatibility locus, and we've talked a little bit about HLA, but what I would like to do is delve into HLA a little bit, talk a little bit about the genetics of the immune system, and then come back to this and try to explain how that leads to diabetes. So we know from genome-wide association studies that there's an increased frequency of particular alleles of these particular genes in type 1 diabetes. Okay? So the picture is a little bit misleading in that the picture here shows an association with these genes, but really what we found in these genome-wide association studies were that there were particular alleles at these genes that were more frequently seen in patients with type 1 diabetes over patients without type 1 diabetes. So let's digress a little bit and recall what these genes actually do and how these genes factor into the immune system, and that will then help us understand the certain aspects of the pathophysiology of type 1 diabetes. Okay, so we looked at this picture before. On chromosome 6, there's a particular region known as the major histocompatibility region. And this is where the HLA genes are located. And you'll hear people talk about HLA class 1 genes, and you'll hear people talk about HLA class 2 genes. HLA class 1 genes result in a receptor that sits on the surface of many cells, such as lymphocytes. And this particular, either of these genes in the HLA class 1 region yields the alpha strand of the receptor protein, and the protein has a little groove, the antigen binding groove, where a foreign antigen can sit. And this allows this foreign antigen to be presented to T cells in order to mount an immune response. Okay? HLA class 2 genes have a somewhat similar function. Here, the particular receptor is comprised of 2 protein chains. The alpha chain comes from one of the HLA class 2 genes, and the beta chain comes from another of the HLA class 2 genes. And in diabetes, we're going to be looking at the DQB1 and DQA1 genes. So here's the DQA1 gene and the DQB1 genes. The A genes result in the alpha component, and the B genes result in the beta component. Together, the alpha and the beta strands come together to form the HLA class 2 molecule, these also sit on the cell membrane of various cells, and together they also form an antigen binding groove, and an antigen, a foreign protein, can nestle into that groove, and once that foreign antigen is nestled in that groove, it can then be presented to T cells in order to mount an inflammatory response. And what's interesting here is that, as you can imagine, the actual amino acid sequence of the antigen groove would determine what types of antigens that HLA protein can bind to and therefore present to the T lymphocytes in order to mount an immune response. So depending on the protein chain, the amino acids here, some people will be able to bind to some antigens and not others. And if you recall, the genetic diversity and polymorphism seen in the HLA region, it's the greatest area of polymorphism that we know of in the human genome. In other words, for the HLA class 1 molecule genes and the HLA class 2 genes, there are more alleles known at these particular spots than there are for any other genes in the human genome. And as it turns out, the bulk of that polymorphism is in the antigen binding groove. So oftentimes, you won't see much variation in this part of the chain, but you'll see tremendous variation here. And this begins to help explain why some people can mount an immune response to some antigens and other people will mount an immune response to other antigens, because it depends on what alleles you have in your HLA genes as to which antigenic peptides can affix to that antigen binding groove to then be presented to the T cells to mount an immune response. So this tremendous polymorphism in the HLA region helps explain why each of us have different substances that we react to. So how does all of this work? This picture tries to explain how all of this works. And so let me walk you through this, and then the next couple of slides are going to zoom in on particular portions of this. And I got to say, out of everything I've studied in genetics, this is probably still the part that, kind of blows me away more than anything, the fact that all of this works. It's also one of the things that helps explain the paradox as to how is it that we have 20 to 25,000 genes, and yet we have potentially millions of different proteins in our body. Okay, and the reason, at least one of the reasons, is the fact that you have, in some cases, multiple gene products combining to form proteins, and so of course you're going to get a lot of diversity there. But another reason we wind up with so many different proteins is because of the alterations in the DNA of these B cells as they go from immature B cells to mature plasma cells. So let's take a look at this diagram in some detail, and then the next couple of slides we'll zoom in a little bit more. And this is probably, boy, if you thought linkage disequilibrium was tough, I think the immunogenetics is probably even tougher, although it's a little bit less mathematical than linkage disequilibrium. So in some ways, it's easier to describe at a conceptual level. If you look at an immature B cell, that B cell has an HLA receptor. Okay, and it has the HLA class 1 receptors, and it has the HLA class 2 receptors, and the actual receptors that are on the surface of an immature B cell really just depend on which particular HLA alleles you were born with. Okay, and whichever HLA alleles you were born with, that determines what HLA receptors are on your B cells, and that's going to be the same for all of the B cells in your body. Okay, and that's not going to change over time. On the other hand, you have other genes that result in immunoglobulin receptors. Okay, and the genes that are responsible for producing the immunoglobulin receptors actually get physically altered in a process known as semantic recombination. Those genes get physically altered, and this mature B cell has now physically altered its DNA. And semantic recombination of a different immature B cell will result in a mature B cell that has a different immunoglobulin receptor on it because it's altered its DNA in a different way. And so you wind up, as your B cells mature and as your B cells go through this process of semantic recombination, you wind up getting mature B cells where the mature B cells differ from one another because they've recombined their genes in a different way. And now you wind up with this mature B cell that has an immunoglobulin receptor, and it's sitting there, for instance, in a lymph node, waiting to be activated. And activation occurs when that mature B cell is exposed to a foreign protein. And the immunoglobulin receptor binds to the foreign protein. And it's estimated that for an arbitrary foreign protein, about 1 in every million B cells can bind to it. So you already have tremendous diversity here in that you have some diversity here in the HLA receptors. You have far more diversity in the immunoglobulin receptors because, as we'll see, the process of semantic recombination yields mature B cells where each of these mature B cells differ in the protein structure of the receptors, whereas all of your mature B cells have the same HLA receptors. So as a result, about 1 in a million B cells can bind to an arbitrary foreign protein by way of the immunoglobulin receptor. The immunoglobulin receptor binds to that foreign protein. The foreign protein is internalized into the cell and chopped up into fragments, and then various fragments of that protein are then exposed on the cell surface in the context of the HLA receptor. Okay, and when the mature B cell presents the peptide or the bound protein fragment, that then can attract helper T cells. And these helper T cells, with their T cell receptors, can only recognize foreign proteins when those proteins are presented in the context of a self HLA receptor. So this allows the helper T cell to first shake hands with the HLA receptor saying, good, I know you, okay, you're one of us, oh, and look at this, you're showing me that we've been invaded by a foreign protein. That allows the helper T cell to activate the mature B cell, and then through a process of somatic hypermutation, further introduce mutations into the B cell's DNA, and that allows the B cell, as it's becoming a plasma cell, to produce immunoglobulin receptors and antibodies that are even more selective against the foreign proteins. So that's complicated. That's complicated, and we need to delve into this in a little bit more detail to explain what's going on. So we're zooming in initially on this interaction between the mature B cell and the helper T cell. So we're zooming in right here, and what we see here is the mature B cell has an immunoglobulin receptor, and that immunoglobulin receptor has been attracted to and is bound with a foreign protein. Okay, so about 1 in a million B cells can bind to an arbitrary foreign protein, and that foreign protein is internalized into the cell and chopped up into a bunch of peptides and then presented on the surface of the cell in the context of an HLA molecule. The T cell, in this case a helper T cell, has a T cell receptor, and that T cell receptor can recognize the foreign peptide when it's presented in the context of an HLA class II molecule. That then stimulates the mature B cell through a process of activation or somatic hypermutation to form into a plasma cell. Before we look at plasma cells, let's just take a little bit of a review about what exactly the immunoglobulin receptor is. Okay, the immunoglobulin receptor is a very complicated receptor that results from 7 different genes, okay? And so on chromosome 14, we have 4 genes. We have the V gene, the variable gene, the diversity gene, the joining gene, and the constant gene. And on chromosome 2 and on chromosome 22, we have genes for a variable region, a joining region, and a constant region. Now, what we're going to look at is, actually we have multiple V genes, we have multiple D genes, we have multiple J genes, and each individual B cell selects one of the V genes, one of the D genes, and one of the J genes, and that's what gives that particular antibody or immunoglobulin receptor its specific characteristics. So this is the antibody. By the way, it might be a little bit confusing. An antibody molecule is the same thing as the immunoglobulin receptor. The immunoglobulin receptor that sits on a B cell can also be secreted. And so the immunoglobulin receptor on a B cell and an antibody molecule are one and the same thing. It's just whether it's anchored in the plasma membrane or whether it's been released to circulate. So these are very complex molecules that sit there on the surface of a B cell, and they are the result of multiple genes all working together. And let's take a look at how this works. First, we have the immature B cell going through a process of somatic recombination to form the immunoglobulin receptor. So if we look here, we see that on the chromosome, we have multiple V genes, somewhere around 80 V genes. We have multiple D genes, diversity genes, somewhere around 30. We have 6 or so J genes, or the joining genes. And what happens is the DNA of the B cell, of the immature B cell, is physically altered. All of the V genes except one are literally spliced out. And all of the D genes except one are literally spliced out. And all of the J genes are literally spliced out. And the DNA in the cell now has one of the V genes, one of the D genes, and one of the J genes sandwiched together. And so the messenger RNA now is unique for this particular combination of V, D, and J genes. And some other immature B cell that's maturing to a B cell will have a different type of rearrangement. And of course, with each rearrangement, you get a slightly different immunoglobulin receptor. And that's the process that's going on here. When the mature B cell encounters a foreign protein, and is able to present that protein to the T helper cell, and then is activated by that T helper cell, the mature B cell undergoes a process of activation or somatic hypermutation. And in that process, the genes in that particular B cell are actually mutated, deliberately mutated within the cell. And what happens is there's all kinds of mutations introduced, and some of those resulting B cells have low affinity for the offending antigen, whereas some of those mutations by chance result in very high affinity binding to the antigen. And those particular cells that have high affinity binding to the antigen are then further stimulated to form into the mature plasma cells. So the somatic hypermutation is triggered by binding to the helper T cells, and then the process is you get these random mutations, and the random mutations will at some point produce antibodies and immunoglobulin receptors that have a high affinity, and that high affinity seems to result in a prolonged interaction between the antigen and the cell surface receptor, and it's that prolonged interaction between the antigen and the cell surface receptor that's thought to further stimulate that particular cell line to replicate, and so you wind up getting a whole lineage of these mature plasma cells that are high affinity for the offending protein. Okay? And that's how this process of somatic hypermutation works. And to me, I find it to be pretty incredible that your germline DNA starts off with just a whole boatload of these V, D, and J genes, and your mature B cells all differ. And even at this point, you now have a pretty good opportunity for your B cells to glom on to a whole slew of different types of foreign proteins, but they're not necessarily connected to those foreign proteins with super high affinity. And from there, then, you wind up having these somatic mutations. And as a result, if you look at the immunoglobulins and the antibodies produced by these plasma cells, you'll see that there's as many as 100 billion different mature antibody molecules that can be produced. So from these genes here to the final product, we're looking at tremendous diversity, and yet also with tremendous specificity. And that's what I find so remarkable. You can have 100 billion different types of antibody molecules being produced in response to specific triggers. What we looked at in the last slide was really the process of B cell development. When B cells develop, they become plasma cells that can secrete antibodies. And those antibodies then can glom on to foreign proteins, or in the case of an autoimmune disease, those antibodies can glom on to self-proteins. But I didn't want to leave you with the notion that autoimmune destruction is always B cell mediated. Sometimes autoimmune destruction or destruction of infected cells can be T cell mediated as well. So what we looked at over the last few slides was the major histocompatibility, or the HLA receptor presenting a foreign antigen to a T cell, and that would then activate the B cells to become plasma cells. In a somewhat similar fashion, you can have an antigen-presenting cell with an HLA class 1 molecule present an antigen to a killer T cell or to a cytotoxic T cell. The cytotoxic T cell, through its T cell receptor, can recognize a foreign antigen being presented in the context of an HLA class 1 molecule, that can then activate the immature T cell to form a cytotoxic T cell. And this plays a role in diabetes. This is also, as we're going to see, plays a role in immunotherapy in cancer, which we'll come back to next week, because we can find ways to identify foreign proteins in cancer cells, and we can stimulate a whole slew of cytotoxic T cells to be produced. Those cytotoxic T cells can then be infused back into the patient, where they can then go attack those cancer cells, and where the cancer cells have antigens that are not found on normal cells, we now have an effective immunotherapy against those particular cancer cells. So we see two types of immune responses, and there's others, but at a high level we see two types of immune responses. One is the cell-mediated immune response that results in B cells being activated and generating antibodies. And then we see the T cell type response, which results in cytotoxic T cells being activated also against foreign antigens. And this picture here was just too groovy for me not to present, even though it's not really a diabetes picture. This is actually an HLA class 2 molecule with the alpha chain and the beta chain, and the protein fragment that we see in here is a gliadin protein. It's from wheat, and this is thought to be part of the pathophysiology of celiac disease. We have the HLA class 2 molecule presenting a little fragment of the wheat protein, and now if a cytotoxic T cell comes along, if a T helper cell comes along, and so we're looking here at a HLA class 2 molecule, and if a T helper cell comes along and then connects to this, it then stimulates this particular B cell to produce antibodies. And if in fact this gliadin protein happens to be similar to a normal protein found in the body, or what's referred to as molecular mimicry, then the antibodies that are produced by that B cell, in addition to being targeted against the gliadin peptide, those particular antibodies may also attack the body itself through this process of molecular mimicry. So this was just such a nice picture that even though this is really a picture for celiac disease, the pathophysiology of the immune activation is similar what we're seeing in diabetes. And so if we want to summarize at least one possible mechanism of the pathophysiology of type 1 diabetes, which we see here, we see you begin with the genetic susceptibility. In other words, you have to start off with the right alleles of DQA1 and DQB1, because if you don't have the right alleles, then you're not going to be able to bind to these foreign peptides. And by the way, exactly what these foreign peptides are that are triggering this process in diabetes are not known. Some potential triggers that have been identified are viral infections, for instance, early rubella infections, early exposure to bovine albumin. And then there was some interesting studies, and I included some references in the notes in the slide deck. There's a Science article from last year that you can see if you grab the slide deck, where it appears as though when we used to think about what are called neoantigens or foreign proteins in cancer cells. We thought, well, maybe what's going on is, some genes are just being activated and they're surfacing proteins that the body's not used to. But it turns out that in diabetes and in cancer as well, some of the proteins that are on the surface of the cell that are serving as antigens to stimulate an immune response are weird proteins formed by peptide fusions. They're not proteins that are the direct result of translation of the messenger RNA, but rather several proteins may have been produced, but because of the altered cellular machinery, one piece of protein was cleaved and fused onto another piece. And so novel epitopes or peptide fusion can result in weird proteins being exposed on the surface of cells to trigger an immune response. And it's thought in part that the immune trigger in diabetes might be some of these epitopes that are formed by this process of peptide fusion. So anyhow, the exact triggers aren't known, but there's a lot of possibilities. These triggers, in the context of the right genetic susceptibility, activate B cells to produce autoantibodies and activate cytotoxic T cells, which then results possibly through a process of molecular mimicry, or possibly not, because you have these new epitopes on the surface of these pancreatic islet cells. You wind up having pancreatic islet cell destruction. The destruction then exposes the immune system to other islet cell proteins, and these may have been islet cell proteins that were not that the body hadn't been previously exposed to, and they may serve as secondary triggers, and you wind up getting this cascade of immune destruction leading to diabetes. And I say that this is one possible mechanism because, of course, not everybody with diabetes has these particular alleles, and there's obviously a lot of other mechanisms at play in diabetes. So with that, let's go over a few questions. If your head isn't swimming too much, or if you're not marveling too much at what the heck, this is really what's going on in the immune system, and this notion of somatic recombination leading to 100 billion possible antibody molecules that are highly specific. So first question, all immunoglobulin receptors are identical within a given individual.
False.
True or false, right? Somatic recombination and somatic hypermutation can result in billions of possible receptors within a single individual, as opposed to HLA receptors, which are all identical within a given individual. Okay. What is the most genetically polymorphic region in the human genome?
HLA.
HLA. Very good. Very good. Chromosome 6, the major histocompatibility region. For a helper T cell to stimulate activation of a mature B cell into an antibody-producing plasma cell, the T cell receptor must interact with the B cell's immunoglobulin receptor or the B cell's HLA receptor?
HLA receptor.
HLA receptor, very good. The T cell only recognizes a foreign protein when the foreign protein is presented to the T cell in the context of a familiar HLA receptor. Okay? All right, good. So let's turn a little bit now to treatment. Okay? And first, let's just look at general treatments, and then we're going to focus a little bit on molecular treatments. And as you can probably guess, I mean, there's gazillions of treatments and there's gazillions of innovations, and any attempt to categorize treatments in any way, shape, or form is going to break down. But it's kind of a useful way of at least conceptualizing the realm of treatments that are out there. So here we can think of treatments spanning anywhere from the mutant gene to the family, with genetic counseling. And where the gene is mutated, there's transplants, there's gene therapies, and we'll come back to gene therapy in more detail when we talk about genetic engineering. We also saw for thalassemia and for sickle cell disease that there's ways of pharmacologically modulating gene expression. And so we looked at using decitabine to increase hemoglobin F in sickle cell disease and thalassemia. Sometimes the treatments target the mutant messenger RNA, and there's therapeutic options to silence the mutant messenger RNA through a process of RNA interference, where we inject something or get something into the cell that binds to the messenger RNA, preventing the messenger RNA from being transcribed, for instance, or resulting in the RNA being degraded. Where the protein is affected, we've looked at this for cystic fibrosis. There are drugs that can be administered to enhance residual function. The lubicaftor and the imacaftor for cystic fibrosis. There's protein replacement. We looked at recombinant factor VIII for hemophilia A. Metabolic or other biochemical dysfunctions can be treated, just for instance with a low phenylalanine diet and PKU. For the clinical phenotype, there's medical interventions, just your basic stuff, transfusions and surgical corrections. If we look at the realm of molecular treatments, by the way, the abbreviations for everything in this picture are here in the slide deck. And this is kind of a big picture view, and even this picture is missing, is missing some of the different treatments. For instance, we'll talk next week about monoclonal antibody treatments of cancer. So even when you wind up having a mutated protein here, you can, if you will, silence the mutated protein by injecting a monoclonal antibody that binds and kind of inactivates the function of that particular mutant protein. But starting from the gene, working your way through the messenger RNA, through the protein, there's a number of different treatment options. And again, this list kind of sort of gives you just an idea of the scope of what can be done. DNA hypomethylation therapy with decitabine has been used to increase fetal hemoglobin synthesis in sickle cell disease and thalassemia. There's also drugs that are used to increase expression of a gene. Danazol, for instance, has been used to treat hereditary angioedema by functioning in the promoter region to increase transcription of that particular gene. If this is the promoter region and this is the transcribed sequence of the gene, these antisense oligonucleotides can bind to particular portions of the gene and can result in changes in the transcribed messenger RNA. They can result in skipping various mutated sections. They can result in jumping over sections. These antisense oligonucleotides have been used, for instance, in muscular dystrophy. We're going to come back to muscular dystrophy when we talk about genetic engineering. We won't be talking so much about these antisense oligonucleotides. We'll be talking about CRISPR-based therapies to get in there and directly edit. One of the challenges with these antisense oligonucleotides and even these short interfering RNAs is trying to figure out how to actually get them into the cell. And so it might be that these are of interest, but the complexity ultimately is leading people to look more at gene modification using techniques like CRISPR. But these ASOs can bind to various segments of DNA. They can induce skipping a mutant exon. ASOs are also used in cystic fibrosis to correct one of the mutations and result in restoration of the normal messenger RNA. Short interfering RNAs bind to the messenger RNA and, for instance, can block it from being transcribed and result in the messenger RNA being degraded. And then when you do wind up having the protein, sometimes the protein is abnormal, and lumacaftor in the common mutation of cystic fibrosis can be used to kind of facilitate folding of the protein, letting the protein work its way through the Golgi apparatus and working itself to the cell membrane. And then when you have this other mutation in cystic fibrosis, the glycine 551 asparagine mutation, ivacaftor can improve the function of that protein. And then, of course, we have protein replacements, like with hemophilia. We have cofactors such as pyridoxine in classic homocystinuria. And then, of course, we also have the monoclonal antibodies, which can bind to these proteins and kind of get them out of the way. And I think for today, that is it. That is it. There was no informatics discussion today because I just really wanted to spend time going over the immunology of the genetics of the immune system. So that's it for today. Thank you, guys. And if we could just spend a few minutes on the evaluation, I would appreciate it. Any questions or any comments about today's session? And I think, Peter, if you're out there, we can stop the recording now, too.